Good evening, everybody. Welcome to this month's webinar for the CMTA Genetics and CMT Q&A with Shawna Feely and Carly Siskind. And just like to say, this should be a really interesting evening. I've had a nice chance to talk to both the presenters, and I think it's going to be a really interesting topic here for us for a webinar. One thing I do want to mention, we want to focus here tonight on genetics and CMT and a Q&A, so questions and answers about the genetics part. We're not going to be talking about all the other things that we would normally talk about sometimes on these webinars, so let's try and focus our questions as we can. For those of you who are new to the webinars, on the right-hand side of your screen, you'll see an area there where there are a place you can ask questions, and we'll be monitoring questions as we go throughout the evening, and hopefully answering those questions as they pertain to any particular slides that we may be on during that time. So with that introduction, I will turn it over, and I hope you all enjoy this presentation. Shawna, Carly?
Thanks, Bob. Thank you so much for that introduction, and thanks to the CMTA for giving us this opportunity to talk to the community about genetics and CMT. I'm Carly Siskind. I'm a genetic counselor at Stanford Healthcare. Sorry, we just changed our names. So Stanford Healthcare. And Shawna, do you want to introduce yourself?
Hi, I'm Shawna Feely, and I'm a genetic counselor at the University of Iowa in the neurology department with Dr. Shai.
So today we're going to go through a lot of questions. Shawna had the opportunity to do some questions and answers with the branch leaders at the meeting in March in Florida, and there were some questions she didn't get to. So we're going to take this time to go through some of those questions that came up then, and some of those are listed here. What I'll say is that in order to give some of the answers to these questions, we do need to do a little bit of background about CMT in general and genetics as well. So we'll start off with that, and then we'll launch into these questions. So the obligatory background on CMT. CMT is the brand name for any kind of an inherited peripheral neuropathy. Inherited means it can be passed on from generation to generation. Neuropathy means there's a problem with the nerves, and peripheral refers to the peripheral nervous system. The central nervous system is the brain and spinal cord. Those are not typically affected in CMT. So things like lifespan, intelligence are not usually part of the picture. What are affected are the nerves that branch off of the spinal cord down to the feet, down to the hands, the periphery of the body, and thus the peripheral nervous system. Both the motor and sensory nerves are affected in typical CMT, though not in every form. So the motor nerves are the ones that allow you to pick up your feet when you're trying to step over a curb, and the sensory ones are the ones that allow your brain to know that the shower water is hot when you stick your toes into it. Proprioception is also often affected, which is the mind's ability to know where the limbs are in space, or in other words, there's the ability to stay balanced when a person is in a dark space or in crowded rooms or on uneven terrain. So when that's impaired in CMT, a lot of people have additional problems in those places. When we're talking about the nerves, we like to compare them to wires because most people have usually seen wires, whereas most people haven't usually seen a nerve. And so as you probably know, there's some kind of a metal inside the nerve inside a wire that a signal is sent down. And then there's some kind of an insulation, plastic or rubber that wraps around that metal and allows the signal to be insulated properly and be sent properly. Same thing is true in a nerve, so that metal part that the metal gets, that's similar to the metal where the signal is sent down, is the axon, and the myelin is the part of the nerve that's the insulation that allows that signal to be sent properly. When a nerve conduction study is being done, it's looking to see in general which part of the nerve is being affected, the myelin or the axon. When a nerve conduction study has a slow speed but a normal strength, that's a problem with that insulation, with the myelin. And so that's called the demyelinating form of CMT. It's also sometimes referred to as the CMT type 1. When the speed is fine, but the strength of the signal is reduced, then that's usually thought as a problem of the axon or an axonal form of CMT. and that's also thought of as a CMT type 2. The background on genetics starts with the cell. So we typically have 46 chromosomes in nearly every cell in the body. The chromosomes contain all of the information our body needs in order to grow, develop, and function properly. And these can be arranged based on their size in the black and white stripes into pairs. So we have 23 pairs of chromosomes, where we receive one member of each pair from our mom and the other member of each pair from our dad. Likewise, when we have children, we pass along one copy of each pair from, to our child, and our partner passes along the other copy of the pair. The first 22 sets of these chromosomes are exactly the same in males and females. We have all the same information, all the same genes. The only place that we differ is in the last pair, which are the sex chromosomes. So this is a picture of a female who has 2 X chromosomes that you can see in the bottom right, versus a male who has an X and a Y chromosome. Our chromosomes, we have 23 pairs of chromosomes with roughly 20,000 genes on them. And just like we have 2 of every chromosome, one from mom and one from dad, we typically have 2 of every gene, one from mom and one from dad. Genes are made up of DNA, which is essentially just a code. It's a series of letters that, when it's spelled correctly, lets the gene do its function. And every gene has a specific function in the body. Most of the genes, when we're talking about CMT, have something to do with that nerve or the energy that goes into making the nerve work.
So Carly was going through some of the ways that we'd help define what type of CMT a person has using nerve conductions, and there's other ways to help define this. So CMT is really an umbrella term. It's a group of disorders, and their symptoms can be similar to one another, but they're actually different types of diseases. And so there's different groupings that we use different subtypes that we use to define CMT. So these subtypes are based on both the nerve conduction studies, which Carly described, as well as how they're inherited in the family. And so we're going to go through the different inheritance patterns. But broadly, there's autosomal dominant type of inheritance, X-linked inheritance, and autosomal recessive inheritance. And using the family history, we determine whether it's dominant, X-linked, or recessive. And then from there, if it's dominant and it's demyelinating, then it's a type 1, as Carly mentioned. If it's dominant and axonal, then it's a type 2. If it's X-linked, it's considered a type X because it's on the X chromosome. And then recessive groups, demyelinating or axonal, so recessive forms of CMT, are grouped into this subtype of type 4. So then when you move on from a subtype and you actually do genetic testing and identify a genetic cause for the specific CMT that a person has, a letter is added. So when you have a number and a letter, then you have a specific known type of CMT. So that letter tells you which gene is causing those symptoms. So type 1 is still a large group of conditions because there's many different causes for type 1 CMT. But 1A is a specific gene causing a specific set of symptoms. And likewise, 1B, 1C, 1D, and the list goes on and on. There's also 2A, 2B, 2C, 4A, 4B, going on down the alphabet. So every time a new gene is found that is found to cause CMT, then it becomes another letter basically on the alphabet. And we now know that there's 90 genetic causes for CMT that are known.
So, Shawna, this is Bob. I have a couple questions on that slide, if we could go back for 1 second.
Mm-hmm.
The first question is, somebody who has a proven CMTX mutation is wondering, is it possible for them to also have like a CMT1 mutation? So should they keep looking, or should they stop once they know they have an X?
Typically, people are going to only have one type of CMT in their whole family. So if they've found a genetic cause, then that's where we basically stop, unless they have very unusual symptoms. So the most common form of CMTX is CMT1X, and that's changes in a gene called GJB1, and that's one of the common causes of CMT. So if someone's symptoms match that type of CMT and their nerve conductions match that type of CMT, then we would, once we found a mutation in that gene, we would stop there and say they have CMT1X.
Okay. And the follow-up question was X-linked intermediate. What does the intermediate mean? What does that define?
Yeah, so type 1 forms of CMT are clearly used when something's clearly demyelinating. So that means that when we do nerve conductions, we're looking at the speeds, and the speed should be 50 meters per second. For people with demyelinating forms of CMT, those are very slow, like usually 20 to 25 meters per second, but they can even be less than 10 meters per second. For axonal forms of CMT, the speeds are okay, but the strength of the signal is weak because of the axonal damage. So we'll still see speeds about 50 meters per second for people who have an axonal CMT. For X-linked CMT and other forms of intermediate CMT, the speeds are somewhat slow, but not very slow. So they're in between 50 and 25. So you can see speeds around, 30 to 40, and that's called an intermediate type of CMT. So there's any intermediate forms of CMT, and then there's also the X-linked forms can show that intermediate slowing, or they can be near normal speeds.
Okay, and the final question, another from the X-linked. There's no subgroup yet for X-linked. It's just type 1.
Well, there are subtypes. So there's 1X, 2X, 3X. It just depends on the different genes that are associated with it. But it's very the most common, and the other ones are very rare. So typically, if someone has an X-linked CMT, they have CMT1X.
Thank you.
Mm-hmm. So one of the questions that people had is, what are the most common causes of CMT? And actually, we've done research on this so that we know that CMT1A is definitely the most common, and that's caused by a duplication of a gene called PMP22. Then the other more common forms of CMT are CMT1X, CMT1B, and CMT2A. And then the reverse of CMT1A, which is a deletion of PMP22, which causes HMPP, which is still considered a form of CMT, even though it has a slightly different name that describes the symptoms, because people have these symptoms that are sort of more transient, but we still consider it a form of CMT. So this grouping is the most common causes of CMT, and for the most part, in terms of patients getting genetic testing, you could do a mini panel with just these tests, and that would pick up the majority of people with CMT.
I think the number is something like 90 to 95% of people who have known genetic mutations have a change in one of these 4 genes. Is that right, Shawna?
Correct, yep. So people who are maybe out in the community and don't have access to one of the centers of excellence, but they're working with a doctor who is helping them get genetic testing, if the doctor doesn't know a lot about how to narrow down what type of CMT a person has, doing a mini panel capturing these genes is an appropriate way of trying to keep costs low and try to get the highest hit rate for yourself. The reason why genetic testing for CMT can be so complex is illustrated by this map here. Like I said, there's 90 currently known causes for CMT, and there's more are being identified, all the time. So through the advancements of clinical genetic testing and research genetic testing, we've been able to identify more and more genetic causes of CMT. There's a thought that we will continue to find genetic causes until we find around 200, and we're only at 90. So there's a lot of genes yet to find out there, and a lot of those are for people who have CMT type 2. So the Inherited Neuropathy Consortium, which is associated with the CMTA and the Centers of Excellence, are really focusing on trying to find new causes of CMT, especially CMT type 2. And of the 90, our group has found 50% of those. So we're very proud of that. So it's very complex when you get to genetic testing for some of the more rare forms of CMT, and depending on the lab and things like that, it can be expensive for patients. So a lot of times working with a genetic counselor can help to sort of narrow the scope, because as you can see, there's genes on all different chromosomes that can cause different forms of CMT.
There are increasing numbers of labs that are doing genetic testing at this point, and this is, I looked yesterday to find out how many labs were doing it. So there's 14 labs now that are doing testing for hereditary neuropathy panels, and these panels can range from 2 genes to 102 genes. Having a person who's able to help navigate that system, such as a genetic counselor or one of these centers of excellence, is really helpful to try to figure out what's the best one to do, because as Shawna said, if we have 90 genes that have been identified as causing CMT at this point, and Mayo's doing testing for 102, what are these additional genes? What else are they looking at? Is bigger always better? Or is something like the Athena and Quest panel, which is 23 genes, which is a smaller panel, but they're all genes that are really well established and known to be associated with CMT. Is that a better way to go? So figuring out, is bigger better, or number of genes that are more associated with CMT, and then how to interpret the test results and how to work with all of these various labs are all really big challenges from the genetic counseling point of view. And so picking a lab and picking the right person to help with your diagnostic odyssey is actually a really important piece of the puzzle.
Yeah, not all labs are created equal, so they have different genes on those panels. They all cost different amounts, and so it's really challenging to navigate through that. And genetic counselors kind of know what to look for to help guide patients through the testing process.
A quick question on this slide that we just saw there. Somebody would like to know, like, how far things have progressed over the past 5 years with genetic testing, and do you guys see that happening, in another 5 years? We have 102 at the top here. Will there be 400 in the next 5 years?
So the first part of that question is how much have things progressed, and they've progressed to an astounding level. When I first started in CMT, there was one lab in the country that was doing genetic testing, and if you wanted to get a panel test with maybe 16 genes, that was being done through a type of sequencing called Sanger sequencing, which is really labor intensive, and it goes letter by letter by letter of the DNA code. And that testing costs upwards of $10,000, someplace between $10,000 and $20,000, depending on what test was ordered. Versus now, the test methodology is called next generation sequencing or massively parallel sequencing, and it can be done by a computer very quickly, very easily, and in huge panels at once, so that the intensive part now of doing the testing isn't the actual running of the sequence, but it's of the interpretation of the results. The changes, I think that to the second part of your question with how things are going to progress in the next 5 years, my guess is that there's going to be more and more technological advances. They're talking about a whole another generation of sequencing coming on board soon. And so there's going to be more being done. But I still hesitate with the is bigger better. I mean, if there's going to be 200 genes total that we think are going to cause CMT, why would we test 400? What else are we picking up, and what else do we not know enough about and can't interpret and get results that are uninterpretable when we're doing these bigger panels? In general, I think technology has gone a lot faster than our ability to interpret results, and that's been a huge limitation in genetic testing.
Okay. And a follow-up again, can you detect CMT with a saliva test instead of a blood test, or does it have to be a blood test?
Some labs can do saliva. I think Invitae is doing saliva in some situations. That's only one I know off the top of my head, but I'm sure other labs are coming on board doing that. They get more DNA out of blood tests, so sometimes you get better. You may end up having to submit a second sample if you're doing a saliva sample, but a lot of labs are getting certification for being able to do saliva.
Okay, thank you.
So when we do genetic testing, I'm sorry, Shawna, is it you?
No, I don't know. Go ahead.
Okay, so when we do genetic testing, we can get 3 different results out of the test. It can come back positive, or it comes back with an answer, and we can tell you exactly what type of CMT you have. It can come back negative, and so there just wasn't a change that was on any of the genes that were looked at that would explain the symptoms. Or it can come back with a variant of uncertain significance. And this is one of the most challenging pieces for me as a clinician. When I get back a variant of uncertain significance, that doesn't mean that it is not disease-causing, and it doesn't mean that it is disease-causing. It means it is of uncertain significance. So that often a lot of times people have to wait for additional studies to be done on the same gene at the same place at the same DNA letter, see if anyone else turns up with the same thing, or other people in the family need to be studied to see if they have it. And sometimes it just takes time and patience to figure out if it means anything. And sometimes we don't know for years and years and years if it is actually something that's of clinical significance. So it can be, for some people, even more frustrating than getting no results or a negative result.
So Carly, when looking at these, if we could go back one slide there real quick, thanks. Yeah. We have someone here who has CMT type 2A, and it's kind of 2 questions to that. First, is it still passed down 50-50, 2A? It's a woman. And second, she has fraternal twins. If one was tested, is there still an advantage to testing the second?
So we will get into inheritance patterns a little bit later in this talk, but just to quickly answer the question, 2A is a autosomal dominant form of CMT, and so a person who has CMT 2A is a 50-50 chance in each pregnancy or for each child. So for fraternal twins, it would be 50-50 chance, and the outcome of one child does not influence the outcome for the other child. So if your other child is having symptoms, then it would be maybe worthwhile to have that child checked, but if your child isn't having symptoms, then don't necessarily need to do anything at this time.
And getting to Carly's point about variants, the MFN2 gene, which is known to cause forms or known to cause 2A, there are variants in certain significance in that gene, and there are also what we call polymorphisms or benign changes that don't result in disease. So MFN2, that particular gene, can be challenging in terms of interpreting the information if the change that was found is not a known mutation that's been documented in families before. So that's where you have to be cautious, because someone might get testing and they find a change and they assume that it causes CMT2A, but that's not always the case in changes that are found in MFN2.
And one more question to follow up on this. someone here has CMT1A, and both of her sons were tested a few years back with the results coming back negative. So, so that would, you know, can it skip a generation is the question, or can they feel pretty confident?
That is one of the questions we're going to get to later.
Ah, there we go. Then perfect.
We will come back to your question. So Shawna, what do we do if the testing is negative?
So yeah, that's a good question. What do we do if testing is negative? Does that mean that that person does not have CMT? No, it doesn't mean that that person doesn't have CMT, because as we know, we have many genetic forms identified, but there are still ones that are out there that elude us. And so a person can have a very large panel done of CMT genes and be negative for that, but it doesn't take away that CMT diagnosis, because if their symptoms, their family history, or their nerve conductions are still consistent with CMT, we would still diagnose them as CMT even though a genetic change is not identified at this time. So
how can genetic counselors help with that?
So one thing people can do is work through a genetic counselor to try to guide them to what the best test approach would be, and then also to lead them down paths where other testing, follow-up testing can be done. So genetic counselors help to guide people through the testing process, facilitate the most cost-effective approach to testing, and identify the most likely way to get a positive test result for patients, as well as review the pros and cons of testing for patients. They can also, in doing this guiding, they can also look for other resources, such as research-based genetic testing or other types of testing that can try to get an answer for families. And that especially is available through the Inherited Neuropathy Consortium, through those centers of excellence, because we have a research project using whole exome sequencing to be able to identify these novel or new causes in families. And so working with a genetic counselor at one of these sites, you would have access to free research-based testing if your family is one that a genetic test or genetic cause has not been identified.
Shawna, what is exome testing?
Exome testing is, instead of looking at the 90 or so genes that are known to cause CMT, we expand exome sequencing so that we're actually looking at all the active genes, so meaning all genes that create a protein. So it's a section of the genome so you're looking at genes that are known to create a protein in the body. And so this 85% of genetic diseases are found in this section of the genome. And so we can screen this large portion of the genome and look for genes that are known to be associated with CMT, but also other genes that could be associated with CMT. We just haven't found them yet. And so in this way, you can screen a lot of data at one time. And what we do is we work with families to get multiple samples, pair that data, and we try to find the genes or the mutations that are located in patients that have the disease in their family. And those mutations should not be present in the people in the family who don't have CMT. So using this exome sequencing process and this new technology is a way to actually identify new causes of CMT.
Okay. So can CMT skip a generation? Can I give my short answer?
Yes,
my short answer is no. But let's go for a longer answer. Shawna, talk to us about autosomal dominant inheritance.
So autosomal dominant inheritance is what we were talking about before with CMT2A. So that's where there's a change in one copy of the gene, but that's enough to cause symptoms in that individual. And so because you only need a change in one copy of the gene in order to cause symptoms for dominant forms of CMT, the partner genes doesn't matter in this case. And every time a person would have a child, they would either pass on the chromosome with the change indicated by red here, or the one without the change. And so for dominant forms of CMT, there's a 50% chance of passing it on for each pregnancy. If someone inherits that change, they would have a 50% chance of passing it on, and they would have CMT themselves. If someone does not inherit the change, they can't have CMT and they can't pass it on. It would end there. So when you look
to the
at
question that was given earlier today on this call about if a person had their children tested and their kids were tested negative, do their kids need to worry about passing it on in the future?
no, the child, if they did not inherit the gene change, they can't have it and they can't pass it on. So when we look at a pedigree, which is a family history that genetic counselors draw up, with a pedigree that has, or a family history that has a dominant pattern of inheritance, the people who are affected are indicated in blue. And so each of those individuals would have a 50% chance of passing it on. So you can see here that only the individuals who have it have a chance of passing it on, so they have children that are affected. The people who don't end up getting the CMT can't pass it on, so those people will never have CMT and will not be able to pass it on, so we'll end there in that line of the family. But really we know that families can have variability in their symptoms, and one of the main causes of CMT, CMT1A, there can be a lot of variability. And so this map just shows that that variability based on the color. So the darker the color would indicate more severe symptoms, and the lighter color would indicate less severe symptoms. So there's a gradient or a variability even in the same family. And even though someone might have mild symptoms, they still have CMT and they still have a 50% chance of passing it on. So we have had families where a grandmother was affected and a granddaughter was affected, but the father in between those two was so mild that he was actually surprised that, you know, he had CMT1A, because you can have such variability. So if someone is, you know, family planning and thinking that they might have a chance of passing CMT on, they may want to see a genetic counselor if they have a family history of CMT, just so they can see what the chances are for sure, and they can get genetic testing to make sure that they're not, they actually don't have it, even if they don't think they're showing symptoms.
In recessive inheritance, which is, as Shawna was mentioning towards the beginning, those CMT type 4s, it's a different inheritance pattern, but this is something in rare situations you may actually end up seeing it, quote unquote, skipping a generation, though I'll show you how it doesn't quite do that there either. In this picture, in the top line, you have two parents. Each parent is a carrier of the condition. So in recessive inheritance, both copies of the gene need to have a change in it that causes that gene to not work properly in order to be affected. If a person has only one change in one copy of the gene, the other copy of the gene is working fine, and it's able to pick up all the slack, and that person does not have any signs, symptoms, any changes on electrophysiology. The person will not know that they are a carrier in any way. In every pregnancy, you pass down one copy of your chromosome, your partner passes down the other. So you can see here that there's 4 different ways that these pairs of chromosomes can be passed on. That a parent can pass down both copies of the chromosome without a change in it, and that person would be an unaffected non-carrier. The person could pass on one copy with the change and one copy without from each parent. And so 50% chance of having a child that's a carrier, just like the parents, no signs, no symptoms, no problems. And then a 25% chance in each pregnancy that parents could both pass down the copy of the gene that does have a change in it, and that person would end up being affected with the CMT type 4. In that case, for a person who is affected with the recessive condition, the most likely scenario is that their partner looks like this. Their partner does not have changes in either copy of the same gene. And a partner could be tested beforehand to see whether or not that person is a carrier. Assuming that person's not a carrier, the person who's affected has to pass down one or other of the copy of the gene with a change in it. And the person who is unaffected would pass down a copy of the gene without a change in it. That all of the children of a person who has recessive form would be carriers. Now, the only way, as I said before, it could quote-unquote skip a generation is if you did have one of these carrier children who then had a partner who was also a carrier, and then they would be back into that previous situation where two carrier parents would have a chance to have a child that was affected. But it isn't a true skipping of a generation. It's the genetics are following along with that plan. This is kind of a picture of what we typically see in autosomal recessive inheritance. You can see a person who's affected who may have one sibling who's also affected, or you could see just one person who's affected in the family total, but if you see siblings and no parents affected, that's a pretty good chance that that person does have a recessive form. Particularly if the parents are related in any way, that also increases the chances that the child does have a recessive form. In X-linked inheritance, the way it's passed down is a little bit different from the form of the autosomal ways. So the autosomal ones are the same, males and females, doesn't make any difference what your gender is. But in X-linked, because we have differences in our sex chromosomes, so that females have 2 X chromosomes and males have an X and a Y, there's differences in the way that it's passed down. For a female who's affected with an X-linked form of CMT, she still has that same 50-50 chance of passing down one or other of her X chromosomes in each pregnancy. And dad will pass on either an X or a Y in each pregnancy. So in every pregnancy, there's 4 possibilities. Each passes down an X, and it's an unaffected female. Mom passes down the X, dad passes down the Y, unaffected male. Mom passes down the copy of the X with the change in it, and then she would be an affected female when dad passes down the X also. Or if mom passes down the X and dad passes down the Y, then this is an affected male. In X-linked forms of CMT, males tend to have more severe symptoms than females because females have this extra copy of the X chromosome that tend to pick up some of the slack. But most females do have symptoms of some form. 90% of females will have symptoms of some form of CMT. The inheritance is a little bit different when it's a male that's affected. Mom is going to pass down one or other of her X in every pregnancy. The dad is always going to pass down his X to his daughters, and dad is always going to pass down his Y to his sons. So a male who is affected with a X-linked condition will always pass down the condition to his daughters and will never pass it down to his sons. It stops. In this picture, you can see that here's a male who's affected. He has 3 kids, 2 daughters and a son. Since he has to pass it down to his daughters, you see they're both affected and cannot pass it down to his son. But females have a 50-50 chance of passing it down no matter the sex of the child. So in each pregnancy, it would be 50-50 chance. What we look for from a genetics point of view when we're trying to figure out if someone's got a X-linked form of CMT versus a dominant form of CMT, where you also see these multiple generations affected, is we're specifically looking to see, are there any sons that inherited the condition from their father? If a son inherited the condition from his father, he cannot have an X-linked form of CMT.
So we do get questions about family planning options, and this is one thing that we do touch on with everyone. These are personal decisions, so everyone makes the best choice for themselves and their family. we just provide information for people so they know that there are ways of preventing passing CMT on if people know the genetic cause. And it's really contingent on knowing the genetic cause, so this is not available for people in terms of having their own biological children if we don't know the genetic cause. So there are multiple different options for people in terms of having your own biological children without passing CMT on. There's testing that's available during the pregnancy, and this can be done as early as 10 weeks. And they would test the pregnancy to see if it's affected by the same genetic cause of CMT in the family. And if it is affected, then people would make decisions whether or not to terminate the pregnancy based on that information. Some people elect to go through that process. Some people do not want to have to be made to make that decision about the pregnancy. So they elect to do a process prior to pregnancy, and this is called Preimplantation genetic diagnosis. So this is where you take the egg and the sperm outside the body and create an embryo. It's the same techniques that is used for couples who can't get pregnant on their own, so it's called in vitro fertilization. And then at the 8-celled stage, you can select one cell off the embryo without damaging the embryo. It can be tested for the CMT that's known to be in the family. And so you would only use healthy embryos that are unaffected by CMT to create a pregnancy from that embryo, so implanted back into mom. And that way she knows she's having a child that will be unaffected by CMT. So there's different costs that are associated with each of these procedures. PGD, or preimplantation genetic diagnosis, tends to be more expensive, and it can be challenging to get it covered by insurance, although we can sometimes get portions of it covered. So sometimes it comes down to cost, what people, determining the pathway that people may use to prevent passing CMT on. There's another test that's called maternal fetal cell analysis that they can test for some genetic conditions, but right now CMT is not one of them. So when you're testing during pregnancy, there's actually a procedure that you have to do, and there's a picture on the next slide that we can go through. The other options that are available for having and growing a family are donation, so there's or adoption. So there's donor options, like such as a sperm donation or egg donation, and those have varying costs associated with those. There's also embryo adoption, so that's when people have done IVF themselves for their families and they're not going to use all of those embryos, so they can donate those to other families who have a genetic disease. And so you would start from having a full pregnancy and delivery. That child would not be biologically related to you. And then there's traditional adoption, and that's when you're adopting a child from the U.S. or from a different country. All of these things have varying levels of fees associated with them. So the next slide shows the testing that can be done during the pregnancy. Again, this can be done as early as 10 weeks. So the process is either chorionic villi sampling, where they take a piece of the placenta that has the same genetic information, and then they would test that for this form of CMT that's in the family. Alternatively, you can test the fluid that's surrounding the pregnancy. You can test that fluid for the genetic cause, and then you would base decision making based on whether it's positive or not. The next slide shows the process for PGD, so that's preimplantation genetic diagnosis, and that's where they take the egg and the sperm outside the body. So this kind of shows that process. So testing is done by selecting one cell off of that, and then they can implant healthy cell embryos that are unaffected by CMT, therefore preventing CMT in the next generation. So these are all what's currently available. These things may change over time, but again, it's contingent upon knowing the genetic cause that's in the family. And we're very aware that there's different viewpoints on ethical issues and things that people have in terms of their belief system. So when we work with families, we're very aware of that, and guide them through these options based on their own background and beliefs and everything like that. So we wanted to leave time for questions. Go ahead.
Yeah, we want to thank everyone for listening, but we do have a few minutes left for questions. If there's anything else that has come through.
Yep, there's a few things here on the board, and first of all, I'd like to say thank you very much. I thought it was great and very interesting. The first question we have here is, if you've had a negative test done, but it was done with saliva, should you be retested? Do you feel that, maybe the blood test is more precise than the saliva tests?
If the report said that they were able to analyze all segments that were asked to be tested, then there's no reason to think that that's an incorrect test. The only thing that I would say in terms of getting retested is that at some point in time when there's more genes that are more reliably to be tested, then it might be a good time down the road to do additional testing.
Yeah, the testing process, they have to be, if the lab offers saliva testing, they have to be certified in a certain way to be able to offer that type of testing. So if their report shows that they were successful, that's a validated report. And so, it's not that saliva is necessarily, not an inaccurate form of testing. It's just that you can get more DNA from blood when you collect blood. So the labs tend to prefer that because, the yield is higher. But it doesn't change the fact that every lab is different just based on what genes they offer and how much they cost and those types of things.
Okay. I got a person here's family history of CMT, but nerve conduction studies showed differences between this person and his cousins, and one's showing axonal, one's showing demyelinating, and he's trying to get his cousins to go forth and get more of the testing done. Is there anything that can be, in families, do you see that happening often where two completely different forms of CMT could possibly cause these different types of symptoms?
Yeah, it can happen. CMT is a fairly common condition, and we do have families that have more than one type of CMT in the family. It's not necessarily the most likely scenario. The nerve conduction studies aren't all created equal, so sometimes getting the same person to do all of the nerve conductions might show something different than having two different people doing the nerve conductions. And it's also very possible that someone doesn't have CMT. There's a lot of different causes of peripheral neuropathy. CMT is just one of them. So it might be that it just is appearing like a familial cause, but it isn't actually one.
Interesting. Okay. And someone, going back to slides earlier, was curious, is there no demyelinating form of CMT type X?
There can be demyelinating forms. It can be slower or intermediate or axonal. It just depends on male or female and other things in the family. But typically, when we have CMT 1X in a family, for example, the person will, like a male in that family member will have nerve conductions around 30 meters per second, and a female may have nerve conductions around 40 or 50 meters per second. So it can be variable even in the same family.
Okay. There's a question here about if you guys know what the percentage of diagnosis for CMT is done through genetic testing as opposed to just clinical work.
A 2011 paper had, I think, 63% of people had a genetic diagnosis. there's a good third of people who have probable CMT, but we don't have a genetic cause for.
And that is somewhat is dependent on the type a person has. So if a CMT person has CMT type 1, our hit rate is much higher. It can be like 80 to 90% chance that we'll be able to get the genetic cause. If someone has a CMT type 2, more of those have been found over the last 5 years, but we still don't know all the genetic causes. So for someone with CMT type 2, they may only have a hit rate of 30 to 40%.
So we have a question here. Dad and grandmother both had CMT 1A, and this person's nerve conduction test came back consistent with that. Is there still value in doing the DNA test?
Oh, I think that's fine. I mean, it's possible. The chances of it being CMT1A is like 95%. So it's possible that that's not the right answer. But I'm sorry, did someone have genetic testing and it was positive?
Good question. Yes, the mother and
okay, yeah, then no, that's a diagnosis.
Yeah, so, yeah.
If someone had genetic testing and then someone else had nerve conduction consistent with it, from our point of view, that is a diagnosis. When clinical trials come out, it may be that you'll need to have your own genetic testing to prove it, but in the vitamin C studies, we didn't require people to do that, just having a first or second degree relative and consistent first or second degree relative with a positive genetic testing result and consistent nerve conduction velocities was enough to say that you have the diagnosis.
Yeah, the only thing is because genetic testing costs have come down, you know, EMGs, getting a nerve conduction can run around $1,000 to $1,500 depending on how complete they do the EMG and the interpretation, and 1A testing is only like $500 at some labs. So, you know, there's a shift going on where some people, if they have a family history of CMT 1A, you know, they may opt for just doing the genetic testing because it's cheaper rather than doing, you know, EMG, and it's also, you know, it's a blood test versus, you know, nerve conductions, so it's comfort level thing as well. But you know, nerve conductions are diagnostic for 1A testing if you have genetic cause in the family.
Okay. I have a question here. If nerve conduction testing shows both myelin and axonal deficits, does this indicate a particular type of CMT, or could there be more than one type involved, or a different diagnosis altogether?
Yeah, I mean, that can happen.
Open question, yeah.
Yeah, it commonly happens. I mean, even with CMT 1A, which we know is a demyelinating form of CMT, you know, the insulation is not there anymore, so the axon actually, you know, kind of breaks down over time. That's the thing that changes over time. So you can have a lot of axonal degeneration that goes hand in hand with demyelination. So that, you know, happens a lot with people who are, you know, older or things like that with CMT. And so interpreting nerve conductions is, you know, it's a skill, and that, you know, a lot of nerve conductions are done poorly and interpreted poorly across the United States. We see a lot of those at the centers of excellence, and so it's really important to have good nerve conductions and have a trained technician who's working with you as well as a neuromuscular expert, so that they can tease out where the primary problem is, where the problem originates from, in the myelin or in the axon. And some forms of CMT, like CMT1B, can cause both. Some patients can have really demyelinating forms of that, and some people can almost look like they have an axonal form of that. So I don't know if Carly has anything to add there, but.
All right, I think that for when we usually talk about CMT type 1 and CMT type 2, we're very like cut and dry, like slow nerve conductions and normal amplitudes, but that almost never happens. And opposite as well. There's some normal amounts of fluctuation that go along. So if you have like a 1/2 decrease in amplitude, it causes a 1/3 decrease in velocity. So there's all these little tricks to the nerve conduction. So the short answer to the question, Bob, is that doesn't mean you don't have CMT. It doesn't mean that there's more than one thing going on. It just means there's normal changes within the nerve within CMT population.
Okay. Well, I think we're pretty much out of time here. I want to be respectful. I know there's a hard stop here. And so I'd like to say thank you very much to both of you for this conversation. I'm sure we'll have an opportunity again to talk in the future. And I'll leave you guys with the floor if you have any final closing comments you want to make.
I appreciate everyone's time this evening, and thanks for joining us on the call. There are resources available on the CMTA website and also on the Inherited Neuropathy Consortium website. So if you Google INCRDCRC, there's a free patient registry that you can sign up for, and that's another way to get involved if you can't travel to one of the centers to be involved in some of the research that's going on. There's questionnaires and things that you can fill out online, but we appreciate everyone's interest and participation. And if you are looking for a genetic counselor to help guide you through the testing process, you can find those on the National Society of Genetic Counselors website or the American Board of Genetic Counselors website.
And I'll just add one little piece, which is that I've been working on revamping a lot of the genetics on the CMTA website. It's going very slowly and piecemeal, but if you have a type of CMT that you don't see on the CMT website under the genetics section, or it hasn't been updated in many years to your eye, send a note to the CMTA and they'll let me know, and I'll start working on your type of CMT as well.